I'm pleased to describe the history of the American Society for Photobiology. Photobiology is broadly defined to include all photobiological phenomena involving non-ionizing radiation. It is recognized that photobiological responses are the result of chemical and or physical changes induced in biological systems by non-ionizing radiation. Photobiology can be divided into 12 major specialty areas. 11 of these are concerned with the absorption of light, and one is concerned with the emission of light, that is, bioluminescence. Photophysics is concerned with the physical interactions of light and with matter at the atomic and molecular level. Photochemistry is the study of the chemical changes that occur in molecules after the absorption of light energy. Photosensitization is the process by which light energy is absorbed by one type of molecule, the sensitizer, and the activated sensitizer then undergoes reactions that ultimately result in the chemical alteration of another type of molecule. UV radiation effects. This field is concerned with the UV radiation photochemistry of DNA, RNA, and proteins, and the biological effects produced by the photochemical changes in these molecules. And this is the area of my main expertise. Environmental photobiology. The different wavelengths of sunlight exert both beneficial and detrimental effects, not only on individual cells and organisms, but more importantly, on the whole ecosystem, where one deals with the effects of light on species composition and productivity. Photomedicine is concerned both with the detrimental effects and the beneficial effects of non-ionizing radiation. And at this meeting, we're working on the beneficial effects. Non-visual photoreception. Light is received by a receptor in an organism to monitor the environment without forming an image, in contrast to the case for vision. One example of non-visual photoreception is the circadian clock, which controls hormonal levels in birds and animals. Vision, as you know, is the photoreception that results in the formation of an image. Photomorphogenesis, the development of an organism can be influenced by the information in the light. For example, causing the germination of seeds and the flowering of plants. Photomovement. To produce photomovement, plants and organisms depend upon the wavelength and the direction of the light striking their photoreceptors. Photosynthesis is, in this case, it is not the information that's in the light that is used, rather it is the energy of the light that is converted to stabilized chemical energy, and this is light harvesting, a light harvesting reaction, while most of the other photobiological reactions that I mentioned require only a few photons to trigger responses. Bioluminescence. For most people, bioluminescence is represented by the flash of a firefly or the phosphorescence that frequently occurs on agitating the surface of an ocean. When I first began studying the photochemistry of the nucleic acids, I didn't know much about photobiology, and I didn't know many photobiologists. And I thought it would be rewarding. Well, I skipped a paragraph. Let me go back. In the United States, prior to the formation of the American Society for Photobiology, ASP, in 1972, the science in each of these 12 subspecialties was scattered throughout different major societies. For example, UV radiation photobiology was a minor section of the Biophysical Society. Therefore, each of these 12 subspecialties in photobiology were isolated from all the other areas of photobiology, even though there are many common features to all of these different specialty areas. When I began studying the photochemistry of the nucleic acids, I did not know much about photobiology, and I didn't know many photobiologists. I thought it would be interesting and rewarding to get together with other photobiologists in the San Francisco Bay Area. So I started the Northern California Photobiology and Photochemistry Group in 1962. In 1969, the Biophysical Society modified its bylaws to permit the formation of specialized subgroups. So I started the Photochemistry and Photobiological subgroup of the Biophysical Society. The one other photobiological group in the U.S. was the Committee on Photobiology of the National Research Council of the National Academy of Sciences. The committee was started in 1952 to serve as the U.S. section of the International Association for Photobiology, AIP. The Committee on Photobiology was mainly responsible for raising travel money for U.S. scientists to attend this international photobiology meeting. When I became chairman of this committee in 1970, I thought that we should start doing something for photobiology in the United States other than just coordinating U.S. travel to these international meetings. However, the committee members were almost unanimous in their lack of enthusiasm about starting a photobiological society. In order to overcome such negative feelings, I performed a considerable amount of missionary work about the benefits of forming a photobiological society. In April of 1972, I brought up the topic again at a committee meeting, and this time the motion was made and seconded to start a new society. We were on our way. Now there were the practical problems of naming, organizing, and funding the society. Writing the Constitution and bylaws. The first national meeting was held in Sarasota, Florida in 1973. There were 240 registrants. This meeting was small enough so that everybody got to meet everybody. Furthermore, there weren't enough papers on each subspecialty of photobiology to keep everyone busy in their specialty. So people didn't stay in their own area, but they listened to other speakers and other specialties, and they learned a lot. And one of the major achievements of this first meeting of the Photobiology Society was that it served as a catalyst for the birth of molecular photodermatology. Dermatologists mentioned the problems that they had and wanted to solve, and the chemists said, oh, we know how to do that. And so many scientific collaborations were started at this meeting and continue today. Now that ASP is much larger, it is very hard to achieve the same level of personal interaction and scientific cross-fertilization, but we try. I did a lot of work to start the American Society for Photobiology, but obviously I did not do it all by myself. I had a lot of help, and thanks to the people who helped, things have worked out for the American Society for Photobiology. The society is now 37 years old. Thank you for your attention.